I should make it to about 15 minutes because I'm going to need your advice on designing clinical trials to improve iron homeostasis in the elderly. So keep that in mind when you hear this presentation. I need your input because it's going to be challenging. So the general goal of us and many investigators is to discover biological pathways and master genes that are causal to aging, and to target these genes with nutritional, pharmacological, or physiological or lifestyle type of interventions in preclinical models, and then eventually in humans, which is always the challenge. So you see the scheme here of what we're trying to achieve. So, of course, one target that looks very promising is autophagy. And we and others are initiating, and some trials are completed, to stimulate autophagy in a variety of conditions. I think this is probably the lowest hanging fruit, and it's going to be very, very interesting to see what those trials are going to come out with. We have trials with metformin, there are trials with rapamycin, and urolithin A is of interest because it stimulates mitophagy. And there are trials ongoing on that as well. Urolithin A comes from pomegranate juice and is converted in the gut. The other area I think is going to be challenging compared to autophagy stimulation is senescence. Again, the goal here would be to knock out these senescent cells. The issues I think are we don't even know how many senescent cells we have at very old age. And the other problem is that the senolytics that are being designed are quite toxic and can have side effects. But again, very promising area, but it's going to be challenging to knock out these senescent cells with old age. But we're involved with designing some clinical trials, and clinical trials have started. Some use quercetin, which is a flavonol, not so toxic, but dasatinib is quite toxic. So, novel compounds are being designed now to knock out these potential damaging cells, which cause inflammation. The biggest sensation is around, NAD, and this seems to be also a relatively easy target to slow aging down, and the trials have been started, and we just have to see if it works. I think there's going to be some challenges there as well, but we know NAD declines with aging. NAD is important for many different pathways. And here you can see the two compounds that are being tested predominantly are nicotinamide riboside. We have 2 clinical trials on this. And NMN, there are a few trials on that because it's rather expensive to make, but these are precursors of NAD. Of course, fasting exercise will stimulate NAD, and probably less promising is to use inhibitors of NAD-consuming enzymes because, for example, PARP is important for DNA repair. Nevertheless, this is, relatively easy, a B vitamin precursor, and we'll just have to see what the trials are going to look like, and we're involved with it. Just one more slide on NAD showing in all these, boxes, NAD is needed for many, many biological processes that are very important with aging. So watch out for those trials. Okay, my talk is about iron. And again, this is going to be a little more challenging, but I'm going to describe to you what happens in animal models in a few minutes, and then what we're seeing in humans. And then I need your input on how we can maybe, improve this iron dyshomeostasis that occurs with age. On the left, you can see iron is important to bind with oxygen and hemoglobin. We also know iron is important in electron transport and through the synthesis of iron sulfur clusters. So iron is again essential. Iron enters from the diet through the gut. And there's a protein called ferroportin. And this exports the iron into the circulation, and it's bound to transferrin. This ferroportin is under control of a master hormone called hepcidin. And this hormone, if it's too high, if the levels are too high, it breaks down ferroportin at the gut and also at other areas, and thereby, kind of impedes with the iron export into the circulation. Hepcidin expression, as you can see here in red, increases in responses to high circulating and tissue iron levels. So if you have high levels, you're breaking down that ferroportin. The problem is right here. Systemic inflammation or infection destroys that ferroportin export protein, and can lead to anemia as well. And again, ferroportin, we'll see a little later on, is also in muscle tissues. So broadly, we're going to see in older patients that they have anemia. But what's also interesting is at the cellular level, we're going to see increases in iron. We know there's mitochondrial dysfunction. Of course, there's many other biological processes that are not working as well, but since iron is needed in the mitochondria, this is important as well. And then there's functional decline. For iron to go into the cell, so this is the iron you saw a little earlier that comes through the gut. It's bound to ferritin, transferrin, and then it's imported into the cell. The cell has different mechanisms to control iron, but what appears to happen is there's iron deposits broadly. There's iron overload, there's lipofuscins, there's pockets of iron as we get older, and this can lead to oxidative stress. So it's not necessarily useful iron. Also keep in mind, if the iron pool increases, typically this receptor shuts down. It says we have too much iron in the cell, it shuts down. And I'll show you that's the case both in animals and humans. Now, iron needs to go into the mitochondria, so I'll show you some data on how mitopharin is altered with old age and how frataxin is altered. And these are, again, iron regulatory protein at the mitochondrial level. This slide just shows you that in skeletal muscle, there's a marked increase, and these are the ages of the animals from 8 to 37 months of age. There's marked increases of iron as we get older, or these animals get older. The red bars are animals that are caloric restricted, and they show an attenuation. You can see the marked increase, again, also here, micrograms per gram of wet tissues in the very old animals, and caloric restriction is reducing that to some extent. Here, same data, different paper, iron levels young, at low at young age, and then increased at 32 months of age. I indicated that high iron levels in the cell shuts down this transferrin protein levels. So protein levels are decreased, as you can see here, between 6 and 32 months old animals, and also the RNA, messenger RNA is declined. We also did some experiments in isolated mitochondria, and we measured the iron levels there as well. And you'll see in a little bit, iron levels are very high in the mitochondria. And then in the isolated mitochondria, we did the permeability, the mitochondrial permeability opening, so the calcium retention capacity test. Again, it's a type of test that kind of sort of tells the resiliency of the mitochondria to take up calcium. So after each addition of calcium, at some point, the mitochondria will open up its pores and will release potentially apoptotic factors. It's a standard test. So the quicker the calcium is added and the quicker the pores open, that's not a good thing. So you can see some examples here of some representative examples of this calcium-induced mitochondrial PTP opening. And you can see in very old animals, after a few additions, 3 additions, the pores open up and you can detect the fluorescent intensity. If you do a control experiment where you add cyclosporin A, which inhibits PTP opening, it's delayed. And again, here's a younger animal, 18 months of age, and it takes more calcium additions to stimulate PTP opening. So what's the relationship? And yeah, unfortunately, these are correlations with iron. But what you can see in the old 37-month-old animals, the highest iron levels correspond with the quickest, the more rapid PTP openings. And I'm just going to talk about the SSM, the subsarcolemmal mitochondria. Again, you can see the correlation here with the mitochondria with the highest iron levels. They open up quicker compared to mitochondria with lower iron levels. And this is an oxidative stress measure, and you can see a very nice correlation with the mitochondria with the highest iron levels. They also have the highest levels of DNA damage. So, what about humans? We published 2 papers in the past 2 years describing some of these proteins and processes in humans. And what we did is we had young subjects, but we also had 2 groups of older subjects. They were about the same age, around 80 years of age. And some were more, functioning better. This is a short physical performance battery score, standing, walking, getting out of a chair. They did a little better compared to a low functioning group. So we also stratified them in two subgroups, the older people, and we also had a muscle biopsy of the younger folks in this study. So if you look at iron levels, you can see if you compare them with young, there's about a doubling of iron levels in the muscle, with the highest levels in the low functioning individuals. We also looked at muscles iron storage proteins like ferritin, and similar patterns were seen. So there's definitely this increase in muscle iron in the older groups, in both groups. So again, go back to this master hormone. What happened to this hepcidin? Again, recall that it breaks down ferroportin at the gut, so there will be less iron coming in. It's stimulated by inflammation. So indeed, the highest levels of hepcidin, so this could be a target for drug intervention, possibly, were seen in the low functioning individuals. Again, remarkable, this is the most remarkable, this transferrin receptor 1. Again, it's responsive to high levels of iron in the cell. And if it sort of detects this in cell culture in vivo, but also in human, it seems to shut it down. So new usable iron is not going into the cell. CYP8, CYP14 are zinc transporters, but they also transport some iron, and they're also in decline, as you can see. What about mitochondrial iron transport proteins and chaperone proteins? Mitoferrin is transporting iron into the cell, into the mitochondria, and here it's used for iron sulfur synthesis. And this part of the complexes in the electron transport chain. It's critically important for that to be functional. So it seems to be, you know, oddly, it's upregulating as if it's trying to find or find ways to get the iron into the cell, but some of this iron here, again, is in deposits. And maybe that's one limitation of our study. We need to have more sophisticated detection systems of finding iron that's really not useful in these muscle samples. But certainly we see that it's deregulated, and mitoferrin has the highest levels in the low functioning individuals. Look at frataxin. Again, this is a very important protein that's important for the iron sulfur cluster synthesis. It's also an iron chaperone or iron storage protein. And we know that people with Friedreich's ataxia, that's a genetic disease. I realize that, but they have difficulties walking, loss of sensation in the arms and legs, and this worsens over time. Again, look at the whopping decline in frataxin. Quite interesting. Maybe a potential target to see how we can, you know, increase the levels in frataxin at old age. But I'm not sure if I'll be able to see it in my lifetime. So, in conclusion, in our studies, we find increased levels of iron in high and low functioning participants compared to young. I thought what was striking was the lower transferrin receptor in skeletal muscle. CYP14 was declined, but not as remarkable. And then frataxin in the mitochondria.
Christian, sorry, I remind you that due to my mistakes in scheduling this afternoon, you have only 3 minutes.
I'm done. I'm concluding. So we're in good shape. So we also looked at, you know, hepcidin. You saw the highest levels in the low functioning. IL-6 and CRP were increased, but they were not higher. You can read it in the paper. They were not higher as compared to the high functioning. They're similar levels, but they were definitely higher compared to young. And yeah, but there's many cytokines, as we know. And then there were higher levels of mitoferrin. One area I didn't talk about, and it's on this slide, is the measure of ferroportin in skeletal muscle. It's quite challenging. So we don't know if the hepcidin is breaking down the ferroportin. We assume so, but we haven't been able to find good antibodies to measure ferroportin. But we suspect this is declined as well. And ferroportin is the only way to get iron out of the cell. So this is another problem we're facing. So you see multiple problems: import, export, and then, you know, hormonal control, inflammation affecting iron homeostasis. I'll stop there for questions.
Okay, great. Thanks, Christiaan. We have about a minute for questions.
Can I ask you something? Sandra Zampieri. Good morning. Good afternoon. I was wondering what was the nutritional status of these subjects? Did you find any correlation between iron and nutritional biomarkers, malnutritional biomarkers, for instance, retinol binding protein, albumin, prealbumin, in the case you test for that? Or do you have any idea of how they can correlate?
Yes, great point. Yes, we did not measure those. And, you know, as you know, nutritional status is just very, very challenging to accurately measure. I agree, that's a limitation. We need to try to do that in a larger trial and see, you know, but again, even if they take sufficient iron or iron supplements, if the receptors don't work, it's still going to be a challenge. And that's what the problem is, you know.
I agree on that, yes. But
And
in any case.
Yeah.
Okay.
Any other questions?
So, preclinically, they're definitely doing chelation in animal models. The challenge there is going to be that some of the chelations therapies they're using may also chelate, for example, calcium. It may have side effects. They are doing, in animal models, they can manipulate all these different proteins and see that, animals do better under ischemia reperfusion of the heart, and they can control iron levels as well. But I'm looking at clinical trials and how, what we can do. There are some compounds like curcumin that can chelate iron. That's from curry, but whether that's going to be sufficient enough, I'm not sure.